Hello students, I am Dr. Shailaja Jadhav, Professor and HOD in Department of Pharmaceutical Chemistry at Progressive Education Society's Modern College of Pharmacy, Nigri, Pune. We have already seen the history and classification of antibiotics. Today we will see the mechanism of action of penicillins. Students, we have seen the introduction and classification of the antibiotics. Now, we shall see the mechanism of action of beta-lactam antibiotics. We know that the beta-lactams act by inhibiting the cell wall synthesis. Now, to understand how the beta-lactams do this, let us see the structure of bacterial cell wall. This figure here is just a representative diagram of the cell wall of bacteria. The bacteria are classified into gram-positive and gram-negative bacteria. The gram-positive bacteria consist of peptidoglycan cell wall and periplasmic membrane. The gram-negative bacteria consist of an additional outer membrane. Now we have seen that the bacterial cell wall is a cross-linked polymer called peptidoglycan. This peptidoglycan helps to maintain the shape of the bacteria. If the peptidoglycan fails to cross-link, then the cell wall will lose its strength and result in the bacterial cell lysis. The enzyme transpeptidase catalyzes this cross-linking process of peptidoglycans. And all beta-lactams disrupt the synthesis of bacterial cell wall by interfering with this enzyme transpeptidase and hence inhibiting the cross-linking process. Peptidoglycan is a chain of carbohydrates composed of alternating units of NAM, that is N-acetylmuramic acid, and NAG, that is N-acetylglucosamine. The NAM units have a peptide side chain which can be cross-linked from the L-lysine residue to the terminal D-alanine link on a neighboring NAM unit. So, the cross-linking takes place between two neighboring NAM units. This cross-linking is done in presence of the enzyme transpeptidase. Before I go any further, I would like to make a disclaimer here that some of the diagrams in this presentation have been taken from the internet, and I give due credit to the professionals who have prepared these. Now, here in this diagram, you can see the peptidoglycan structure consisting of the alternating NAM and NAG units with the peptide side chains. You can also see where the penicillins inhibit the cross-linking. Penicillins inhibit this enzyme transpeptidase. When penicillins covalently bind to the active site of the enzyme, inhibition of cross-linking process occurs, and this inhibition is irreversible. So we see that the beta-lactam antibiotics inhibit the last step in the peptidoglycan synthesis of the cell wall. Inhibition of transpeptidase leads to a weakened cell wall and bacterial cell lysis occurs. Penicillin possibly acts as an analog of the alanine portion of the pentapeptide chain. However, the carboxylate group that is essential to penicillin activity is not present in this portion. Here you can see more clearly how during the cell wall synthesis, the lysine of 1 NAM unit cross-links to D-alanine of neighboring NAM unit, which happens in the presence of the enzyme transpeptidase. Here you can clearly see that penicillins act on transpeptidase. Another theory for mechanism of action of penicillins is that the penicillins mimic D-alanine due to the structural similarity. Normally, during the cross-linking of peptide side chains in bacterial cell wall, the peptide side chain of 1 NAM unit binds to the serine of transpeptidase enzyme at D-alanine and releases one molecule of D-alanine. Another peptide side chain on neighboring NAM unit at glycine binds to the D-alanine of first peptide side chain and is released from the enzyme, thereby forming a cross-linked product. But when penicillin comes in contact with transpeptidase enzyme, it covalently binds to the serine of the enzyme, thereby blocking the enzyme, because of which the peptide cannot bind to it. Hence, the cross-linking is inhibited. Here, you can see what we have been talking about, the similarity between the beta-lactams and the D-alanine structure, which is part of peptidoglycan cross-linking. Penicillin mimic the acyl D-alanine. But interestingly, 6-methylpenicillin, which is a close analog, is inactive. There is one more theory about penicillin mechanism of action, which says that penicillins also act as umbrella inhibitors, which means that they block the access of the peptide side chains of the peptidoglycan to the enzyme transpeptidase by covering the active site and prevent the cross-linking. So now we have seen the various theories of mechanism of action of penicillins by which it inhibits the peptidoglycan synthesis, that is either by inhibiting the cross-linking of peptidoglycans, or the second theory is mimicking the D-alanine, and the third theory is by causing umbrella effect. I hope you have understood it all. Thank you.